Welcome back
Hello.
to yet another episode of Bio to Bedside. We appreciate you spending some time with us today. Today's going to be an interesting one, isn't it going to be, Jeff?
Thank you, Ben.
Today's going to be fun, isn't it? This one, I think, is a bit closer to my heart maybe than it is to yours, Jeff. Jeff is more the cell biologist, and I'm a bit more the gene therapy nerd. But today, we have the opportunity to speak with Genethon. Genethon is really unique in a lot of different ways. First of all, over the course of their experience as an organization, they're a non-for-profit, and they have directly touched or been involved with 15 different gene therapy trials and products, which is wild inside of this space. So they've been around for a long time, and we have an opportunity to speak with them today. So without further ado, we will bring in Angela. Angela, welcome to Bio to Bedside.
Hello, thank you. It's a pleasure to be here.
Nice to have you. Can you tell us a little bit about what your role is within Genethon? We're really excited to get into this conversation of just what you guys are doing inside of the gene therapy space. There's lots to get into.
Yes, of course. So I'm a head of business development and partner in a Genethon, and I've been here since 2019. So it's been a few years now.
Right on. Excellent. And what's your background? What brought you into Genethon, right? And a non-for-profit that's focused on gene therapies primarily.
Yeah. Sure. So I'm a pharmacist by training, and I've done a PhD, and I work a little bit in science before decided and maybe I needed something else. And so I did an MBA and I joined Sanofi and I worked.
Join the club.
And I did a little bit of commercial operation, and then I joined the business development team at Sanofi. And that was an interesting role for me because it's where you can actually mix interestingly business and science. And so be close to the R&D teams, but still dealing with the business matters at the same time. And so I've been there since now the last 15 years in business development. Until then, I decided that even for my next phase of my life, I really wanted to experience what it is to work for a company that is actually non-for-profit and attached to a patient association. And I'm really happy about that, and I don't regret at all the move.
Excellent.
So I don't want to jump into it maybe right now. Maybe we can kind of circle back to this, but I'd love to know the difference in dynamic between moving from a huge pharma company like Sanofi into a nonprofit company like Genethon, and like what the differences are behind the scenes, maybe, or there's a difference in environment. Like, did you do that intentionally? You mentioned you maybe you sought that opportunity out.
Yes. Yeah, that's a good point. So I had for some time the desire to leave. So I love the idea of contributing even tiny little bit to bring medicine and therapies to patients. And so that was it's been always the mission of my life. But it's the environment on a big pharma company is also that you need to be very careful of return on investment. So, on your bottom line of your earning per share, on the market, etc. Despite the fact that the mission remains the same, but the environment is the environment of a company that needs to be profitable, which is very reasonable given the cost of bringing therapies to the patients. And so at some point in my life, after 20 years plus in that environment, I really felt the desire to experience something else. And so I jumped into this new adventure. There was Genethon, which is a non-for-profit created by a patient association, and where the board of directors are families of children with rare, very often severe and terrible diseases. And these families don't stop in front of anything. They push every type of barriers. No is never an answer. Impossible is never an answer. So that kind of energy, mission, values, it really becomes the reason why you go to work.
Nice.
Is
Excellent.
Is that common to have like basically patient advocates on as the board of directors?
No. I think we are really unique. There's some other maybe example, but no, probably as developed and structured as we are. So the IFM Teleton, just to give you a little bit of background, if that it's okay, was created. Is the IFM Teleton is the equivalent of MDA in the US. So it's the Patient Association for Muscular Dystrophies. And was created in 1957 by a mom of 7 children, 4 of which had Duchenne. And that kind of, so it became bigger and bigger. Today is one of the biggest patient association overall in Europe. Mm-hmm. And it's, they launched the Teleton. The Teleton was really copied by the MDA US-based Teleton, the Jerry Lewis Teleton, in 1987, with the idea to say we need to do something now. We need to. So we've been, helping the families to cope with this horrible disease. They are so rare, so they're under the radar screen. So we need to do something. We need to really change here the equation and try to find therapies. And so 87 first Teleton, it was an incredible success. And in 1990, it was decided to create the first institution, the first lab dedicated initially, it was Genethon, dedicated initially to the genome mapping. There was no, the human genome was not completely understood yet. So this was the first step, because understanding the genome, it means also better understanding the diseases behind when something gets wrong. And so the first publication of the human genome in Nature was by Genethon. So we're very...
You're famous. What the heck?
Yes, we're famous. We got the cover of Nature.
Roll that back for just a second. Say that again.
I didn't realize that. That's crazy.
We were on the cover of Nature. And so we became very rapidly, in fact, a known institution, known research institution. And then it was in 1997, again, pushed by the families, that we started to work in gene therapy. So 97, we are really at the very, very, very beginning of gene therapy. Initially was, and then a couple of years later, we started to already work on bioprocess, which we understood immediately was going to be one of the bottleneck on the development of gene therapy. I can go on forever, by the way. At some point, you need to stop.
We have an hour.
So let me just finish this story. And 97, we started first on a lentivirus ex vivo, and that's where the first product in, especially neurodeficiencies and in blood disease, that came out, and the first clinical trials where we were involved. And we started also quite rapidly moving to AAV, and to work on a specific disease that's called spinal muscular atrophy. This disease is relatively common, unfortunately. The type 1, the most severe form, it's terrible. Children are born with a mutation and they don't live beyond 2 years old. And at that time, I was speaking about many, many years ago, we tested an AV9. So, gene therapy, I don't know how detailed I have to be about gene therapy, but the gene therapy today in vivo is a viral vector with the gene that is the issue, the healthy gene inside, as there's nothing better than a viral vector to deliver gene in the body. They were designed by nature for that. And so we were the first to start studying this particular disease by injecting an AV9 with the gene that was the issue in the disease. And we discovered the AV9 could cross the blood barrier. We were the first to do that. We filed some patents, and we demonstrated in animal models that an AV9 with the gene that was lacking in the SMA patients was actually making the animal model healthy. And that was the origin of what became eventually Zolgensma SMA, that today is commercialized by Novartis. Is a wonderful success because those children now live and they celebrate several birthdays. So.
When was that approval? What year was that in?
So the approval of Zolgensma SMA was in 2019.
Okay.
In the US. And
was it among? I mean, like this might be super ignorant, so you can slap me later. Was it among the most the earliest gene therapies to be approved? When was
The
the
very
first gene
first, the very first was Luxturna, 2017, for an
Okay, yeah.
ophthalmology for an ophthalmology disease by Spark Therapeutics. That now is part of Roche. That was the
Oh,
first.
dang it. I should definitely know that. Okay.
I'm
making a note of that one.
So now I pause, and that's the story of Genethon. So let me also say one thing that is important. We are not for profit, but we are structured like a biotech. This is important. So we have 200 people in Genethon. We are based in the south of Paris in France. We have a quite large research team with preclinical platform, with imaging, a very nice imaging lab to better understand, tissue, and especially for the muscle, it's very important. We have a CMC team, so we do all our process internally. And we try to improve that process continuously, and then we have a clinical team. So we are non-for-profit but biotech, and this is quite unique in the ecosystem.
Yeah, we had on the name is escaping me now, but we had on the telethon from Italy. 2 months ago, and I remember we had I think Celesti. He was the guy we had on from there, and he said kind of the same thing. We're a nonprofit, but we're structured like a biotech company, and they have their own internal R&D and all kinds of stuff. So I'm not super sure how common that is in the U.S. or in North America, but I know 2 of them now in Europe, both in Italy. Or I guess you're from Italy. Genethon's not Italian. Genethon's in France.
Genethon is French.
Okay.
And but this whole I think we have a lot of Italian working at Genethon, and the 2 countries have in fact a lot of expertise now and a lot of talents in gene therapy. Incidentally, a little bit like probably catalyzed by these 2 institutions.
Yeah.
This is so this is so off topic and non-scientific, but Europe is so cool like that. North America is not like that. I go to Canada once a year to see Ben, right? I live in America and I stay in the United States. But it seems like there's, you're Italian, you work in France. Are you based in Italy or are you in France?
No, I'm based in France, been in France for
See, I'm just I'm in Virginia and I stay in Virginia and I never leave, and when I do, it's a big deal.
Right on. So this is so first of all, Genethon has been around forever. You've had an incredible amount of influence inside of our field, both in terms of just the amount of clinical products that you've touched, but then also really successful ones. The most successful gene therapy of all time by far and away has been Zolgensma. And so just the fact that you guys have had that level of influence inside of the field really speaks volumes to what I think will be a really exciting conversation today. And so, folks, if you have any questions for Angela, feel free to put them in the comments. We've already got a couple already, and there's just one from, I hope I don't mess up your name too much, Rabaya. Apparently, Angela, you 2 used to work together at Sanofi in BD, and so she just is giving you a little shout out. Thanks for coming, Rabaya. Hopefully I didn't mess up her name too badly. But then there is a second one here just from Christopher. Before we jump into the structure of Genethon, I know we've kind of touched on it a couple different ways. There is a question here from Chris, which is, no is not an answer. Love that. Does the non-profit status make approval barriers easier? This is good, because as we kind of get into this idea of how Genethon is structured, this is a key component, right? In the way that you are structured, does this give you, since it's more patient-centric and you've got more patients involved in the entire process all the way up into leadership, does that in any way influence or give you an advantage when it comes to interacting with regulatory bodies?
So we haven't had, we obviously have experience with the regulatory bodies with regards to clinical developments. We have 2 program today in one in phase. We have 2 program in phase 3, and so we know very well the regulatory bodies. We have not had us ourselves an experience of an approval. So obviously Zolgensma was approved by Avexis and then Novartis. And so, but it's difficult to say, right? I mean, the regulatory bodies are kind of unbiased generally for that. Obviously, we are close to the patients. We are the patients in a way, if you allow me to say that. So when we design clinical trial, and we're going to talk a little bit more maybe later when we touch upon the Duchenne trial, we really want to make sure that our design is done in a way that once you get the result, is meaningful in terms of what is the impact to the disease. Mm-hmm. And so this should generally help in terms of getting through an approval.
Gotcha. Okay, so just to back up a little bit with regards to just the way that Genethon is structured, not for profit, structured like a biotech company. What is how is the business set up? In terms of revenue, is it entirely based off of grants and donations, or is there also a component, right, where since you're involved in some of these very early stage trials, there's like the ability to help with getting royalties from programs that you send off? How does that all work?
Yeah,
how do you kind of gas to run the car?
It's a little bit of everything. So obviously, when we started in the 90s, it was entirely financed by public generosity through mainly the part of what was raised through the Telethon. As we became larger and more structured and programs started to mature, we also started to have a partnership and license to pharma. Sometimes it makes sense when you have the right partner to actually let the partner to develop your science into the final product. So obviously, Zolgensma is an example, but also we had we have part of revenues coming from licensing and partnership and grants. Absolutely, we have a nice and quite developed grant office. So we try to, but I mean, you touch upon an important point for a non-for-profit driven by patients. Is when I'm asked as a business development person, what is your financial needs? And I don't have a number. It's the more we have, the more we can do. So we are not raising a Series A for thought number of millions to do X, Y, Z. No, we are really constantly looking for opportunity to raise financing to do even more.
Gotcha.
So can you speak on that though? Because even that kind of like scratches something in my brain. What do you mean? Is that sounds kind of like a university almost? Like you're just always like, do you have people who are dedicated to just innovating? Like what do you mean you're always looking for money to do things?
No, in the sense that we don't have a fixed budget. So we don't, because we have, I don't know, typically a fixed number of products that we want to advance, for which we need a certain number of a certain financing to go from A to B and then from B to C. So we have our program, of course, but the more we have, the more we can do. That's what I mean. Yeah. We are employees of Genethon. We are structured like any private organization, but we work on some diseases that are so rare that you have like few patients a year. So we will never be able to partner in a classic way to a pharma. Right. And so it's for that we need to really find the alternative way of financing. And I think that there are so many rare diseases that we're only scratching the surface in terms of what we can do collectively.
So I know like in the US, we have the voucher program. I'm not super sure how that works, but in my head, it's pretty much like if you get an approved therapy for a rare disease, you can get a voucher and you can sell it. I don't know. You make money. Is there something very similar in the EU, or is it just based on sales?
Yeah, in the EU, you have some other incentive to try, because this is is done by the FDA, and it was actually expired, and it went into the whole difficulties of getting back approval in the US for various shutdown and different issues that happened. And it was reapproved in January. Thanks also for the lobbying of many patient association and families. So back again in the US, we don't have it in Europe. No, we have other incentive to incentivize, to promote development in pediatric indication. but we don't have the PRV. But as a European company, we could file an IND developing the US. One of our products is actually currently in development in the US. We have opened up IND, and it's one of the two that we presented at the MDA conference. And so this is something that we could, we're not there yet, but this is something that could be, interesting to explore.
Yeah.
Awesome. So I guess the question would be, you've been able to help support or be directly involved with, at least 15 different gene therapy clinical trials. What is kind of the process, the decision-making process that makes you choose to go after one candidate versus perhaps one where you're like, well, that might have to wait until later, or we're going to have to push that off for a bit? What's that process for actually taking on a gene therapy candidate?
Yeah, there's a bit of history there. So when I arrived at Genethon, there was already set up a certain number of areas where we were working on. So we have a strong footprint in dystrophies, myopathies, and neuromuscular diseases. So we also have an interest in, we've been since many years in the liver. So these are the two main therapeutic areas: liver diseases and neuromuscular dystrophies. There's hundreds of different diseases in this space, so we cannot do everything. So it's a mix of where there is the expertise of our teams, where there is the needs, etc. So we have, when I arrived, we had the pipeline was already quite stabilized, I would say. And as I said already, our reason, the very original reason of creating the patient association and then us was linked very tightly to Duchenne muscular dystrophy. Mm-hmm. So we've been working in Duchenne for many years, and today we have a program in phase 3. Mm-hmm. And in addition to that, we have a team that's been historically very well implanted into another family of dystrophies called limb-girdle muscular dystrophies, a little bit different from Duchenne. It's a family of different dystrophies because there are different mutations. And one of our team leaders has been working on this family of disease for many years. She's very known internationally. She discovered some of the genes that were mutated. And so there's a very strong expertise. And we have 3 products today that are in advanced development. So it's a mix of unmet need, expertise, and certainly a big focus on the muscle and the liver.
Gotcha. So yeah, have you in terms of muscle and the liver, have you guys seen any of the stuff that's come out of Dino Therapeutics, where they've got this BN8 capsid that can transduce into muscle, and it's 25 times less of a dose compared to something like the standard Duchenne gene therapy that's on the market? How does that work when you're thinking about the technology companies as well? How can they come in and collaborate with Genethon?
Yeah. So we do have, talking about capsid, so we really have an AAV, we are an AAV company primarily today. We are thinking about also starting to work on non-viral vector, but we're mainly AAV. And so obviously we have also developed some internal expertise in generating novel capsids. We do have some interesting capsid in the muscle as well. We know very well some of the big players in the capsid. We have conversation with them, but this is an area where we're very active, and we have a few capsid where we have managed to decrease, because the problem with AAV is that they first go to the liver, and so the liver is the first target the first organ to get hit. And then sometimes it's the reason why some of these therapies had unfortunately some issue with the liver toxicity. So we have some capsid that we managed to target the liver by 1,000 times versus the natural serotype, and increase the muscle by 20 to 30 times. So we have interesting technology around the capsid as well. Yeah.
Fantastic. Super cool. Awesome. So you mentioned, obviously, and one of the things we want to spend a decent amount of time with you on today would just be the data that you guys have had so far. We first got connected a couple weeks ago because there was some very positive, exciting data that came out of the University of Florida with a trial that you guys are helping with. Maybe we can start there, but obviously we want to get through that bolus of just really exciting things that are coming down the pipeline for you guys, especially as it relates to gene therapy. I feel like the gene therapy the gene therapy space could use a little bit of good news. So we can focus on some good news now.
Yeah. So in fact, that's right. We got connected because we went to the MDA, the Muscular Dystrophy Association conference. By the way, a great conference there was this year in Orlando, and we had the opportunity to present 2 sets of data related to 2 different products. So the first that you mentioned is one of the limb-girdle muscular dystrophy product we are developing internally. This is called, it's a disease that's called gamma sarcoglycanopathy. It's due to the fact that you're lacking a protein that's called gamma sarcoglycan. This is a protein, is a structural protein of the muscle. So the children that have this disease are born without the gene that can allow the body to produce that protein. So muscle become weak. Out of all the limb-girdle muscular dystrophies diseases, there are, I don't know how many mutations exist. This is probably one of the most severe. And so the children, they generally are diagnosed quite early. They lose their ambulatory, so they cannot walk anymore when they're teenagers, and they have often cardiac issue, and there's an early mortality. So we have, as I said, one of our lead scientists, Isabelle Richard. She's been in the field of limb-girdle since probably all her life. And so she has, thanks to her, we have developed a gene therapy, which has the viral vector and the gene that is able to correct the problem and get the body to produce that protein. So the story is interesting in not just because of the science, but also because of how we ended up at University of Florida. So we pushed this product. As I said, financing is often an issue, and we cannot do everything. And sometimes we have to slow down some of the products, trying to find the right financing, whether it's a grant, whether it's something else. Mm-hmm. And so we had brought this product up to the point that we were ready to file an IND. So we had all the preclinical package, the talks. So we had manufactured the products, and then we connected to a foundation in the US, the Dion Foundation, that has where the family created the foundation. They have 2 children, and the 2 children have the disease, and so it was a very nice collaboration, and they allow us to help us in filing the IND. And to start the clinical trial, which we decided to do it at University of Florida, where we have one of the key investigators with the strong expertise in gene therapy. There is Dr. Barry Byrne. They did a lot of trials in gene therapy.
Shout out Dr. Byrne.
And so we started with the idea to have to test 2 doses, as you often do in gene therapy. A lower and then a higher, and we started to treat ambulatory patients, so the disease is not too advanced. And so we treated so far 4 patients with the first dose, and what we published at the MDA was the result on the first 2 patients. And I mean, it's been defined. So it's early days, of course. It's early days. And what we show is that, first of all, the important point, like 9 months after you treated the patient, is, do you see in the muscle the protein, right? You do a biopsy, you analyze your muscle, can you see the protein? And what we found is that we saw 90%, 90, of the fiber that could express the protein. This is very rare.
Okay, that was my question.
To see in neuromuscular diseases. So the other point that we saw was the fact that you have different biomarkers. Biomarkers are interesting in the early stage of the post treatments, right? When the functional improvement might take a little bit longer. And one of the proteins that we measure in all this dystrophy type of trial is called creatine kinase. This is a protein that is increases in the blood when the muscles start to degenerate. Mm-hmm. And it's very high typically in children with dystrophies. And for these two children treated with our product, we saw a decrease by 90%, 90. So the almost normal level. And so this is a very, very encouraging sign. On top of that, we saw some video of the children that they were treated even relatively few months after. We saw them that one of the typical signs of the disease is takes almost a minute for a child around 7, 8, 9 years old or 10 to raise from the floor. Okay. And with the treatment, the child was able to raise in 20 seconds. So and this is one of the functional, by the way, raise from the floor is one of the functional outcome that you measure in this type of diseases. So it's early days. It's very safe, of course, by the way. This is important. The 4 patients are doing very well, and watch this space because we're going to publish probably more data at the upcoming ASGCT, the American Cell and Gene Therapy in May.
In May?
Yeah.
Yeah.
Oh, wow.
ASGCT.
Okay, cool.
In Boston this year,
in
Jeff.
Boston, exactly.
Maybe you should come this year, Jeff. Lots of good stuff happening there, Jeff. Angela, you're coming in the middle of an ongoing conversation.
Yeah,
right.
It's a good idea to come to ASGCT. So this is another point on the pros side. Awesome. This is fantastic. Okay, yeah.
And by the way, this was all the first dose. So when you have that kind of target, it's called target engagement. When you see that amount of protein that is already generated, then you say there's probably no need to go to a second dose. You're kind of set up. We'll see what happened in the other patient, of course. So this was what we did for this particular type of LGMDs. And then the other set of data that we presented was on Duchenne, on the phase 3 data that we have on Duchenne. So I don't know if... maybe we can speak about that.
Absolutely. Yes.
So I don't know if
I know exactly what you're going to click on. If Rye's going, I've got to go. Yeah. If not, Ben's going, who cares? Rye's going, I got to go.
Okay, so Duchenne is another type of dystrophy, and it's much more common. It affects several thousand of boys. It only affects the boys in the US and in Europe, and obviously it's just talking about US and Europe, but it's everywhere. And so in this case, the disease is due to the fact that we are lacking the gene or we have a defect in the gene that it's able to get the body to produce a protein called dystrophin. It's a very long protein, the longest gene that we have in the body. And the dystrophin is essential to maintain an healthy muscle. So if you don't have the dystrophin, you have already issue to walk. So a boy with Duchenne will never be able to walk properly and rise the stairs alone. By 12, they're on the wheelchairs, and then they start having problems with your diaphragm, your heart, and so you have early mortality. And so the problem of Duchenne is that the gene that is able to make us produce that protein is too long to fit it as it is in the AAV, in the type of viral vector we use. So the strategy has been to try to see if we can truncate, so just reduce, get a reduced protein that still has the functional domains that makes it do what the dystrophy would have done. And there's some rationale to think that this would work, but let's maybe not go too much into the details. So we've been working on this approach for some time, and we have today a gene therapy with a viral vector, a very interesting viral vector that is AV8, that is very known, has been studied in many clinical trials to go well to the muscle. And we have a promoter. It's very good for the muscle, and what we think is the ideal sequence to get the right piece of dystrophin to be functional. And so we've been doing a dose escalation here as well, and we managed to find the most effective dose to be relatively low for neuromuscular disease is 10 to the 13 viral particle per kilogram. This is still a huge number, but it's lower than other approaches similar in gene therapy in Duchenne. They are all above 10 to the 14. So and we think having that kind of dose, 10 to the 13, for a disease where you need to, it's important in terms of safety. And also there is some advantage, obviously, in terms of potential manufacturing cost. So what we published MDA was the complete 2 years follow-up of the phase 1/2 of the children that were treated for that dose. They were in phase 3. We started the phase 3 last year. And so what we saw is that we had a very, very strong impact on what is the functional, the measurement of the functional score. So in order to understand if this is effective or not, there is for Duchenne a set of measurement of the way the patient walk, raise the stairs, raise from the floor. There's all composite in a 34 scale score. So 34, you're perfect. 0, you cannot do anything basically. And so this is accepted by the medical community as the score. It's called North Star Ambulatory Assessment Score to assess ambulatory patients with Duchenne. And so we have managed to achieve, after 2 years, a difference of 9 points, which is huge. 9 points versus patients that were not treated. And it means that basically, if you see the curve, the score is not moving. So the disease is not evolving anymore. Sure. But this is very medical. And what is amazing is to see the children, because this was obviously an open label. And so one of the child that was treated, I think he was 7 when he was treated, so today is 9. Don't forget that 9, unfortunately, is a time where you really decline very dramatically your ability to walk. By 12, you're on a wheelchair. So it's 9, and last summer, he managed to climb the 650 steps of the Eiffel Tower.
Okay, first of all, I couldn't do that.
Exactly.
I'm out of shape. But
and
still.
he was running up the stairs. I'm shivering when it's amazing. So this is at the end count, right? And then you translate time, measuring a score, and seeing it's true. The disease is not evolving, and is actually probably in this particular case is getting better. And there are other aspects. So we saw as well a very good expression on the microdystrophin in the muscle. And here as well, we saw the famous biomarker, the same creatine kinase, the one that is a sign of there is a damage in the muscle is getting degrading. And in this case, we saw a difference of 70%. So nice. The children are getting normal. There is also an interesting aspect of the evolution of the disease, is that because you don't have dystrophin, you create a very strong inflammatory status in the muscle. It becomes fibrotic, and that's why you cannot contract anymore. And this is why you end up on a wheelchair, and eventually you lose the use of your upper arms, etc. So you can measure the evolution of the fibrosis by an MRI, and in the MRI, if you see that your treatment is you measure the fat fraction. Let's not get into. You measure something in the MRI in the muscle that says there is high level of inflammation or low level of inflammation, and generally to get a meaningful result, you should have at least a difference of 2%, and we saw a difference of 18%.
Oh gosh, okay, so it's working.
So there is much less inflammation, much less fibrosis, and that's why I explained probably how the fact that the muscle is still able to allow this little boy to climb the Eiffel Tower.
Do you have any idea of the durability of those results, or is that relatively what is the time frame of that?
So here we are at, one patient is 3 years, and so it's early days, but we do have some interesting, there is a form, it is a dog that is a golden retriever that suffer from this type of disease is a good model for Duchenne. He has the same lack of the dystrophin. And so the golden retriever with Duchenne is a model that we used in the past and during the preclinical development. And a dog is with the golden retriever with this type of disease has problem to walk, it cannot jump, and again, lifespan is very short. So we treated a dog with our product, and we have some of them. There was kept, and he lived 10 years or 11 even. So which is the natural life of.
I was going to say, like, some dogs don't live that way and they're healthy.
And so this is an interesting indication of long term. Most of the preclinical are done in rodents. So there's a.
So that's actually interesting that they can do it. There's golden retrievers that get it naturally, and you can use them. It's kind of like two birds with one stone, where you're treating somebody's dog, it's their pet, which is good. And also you can get.
This is specific colony that we have. There are other dog model for other myopathy that exist. This is specific colony that we have near.
And to be clear, you guys don't give the dogs Duchenne; they just have it naturally, correct? Okay, We had at Roswell Park, we had a colony of woodchucks because the woodchucks naturally they have like some kind of hepatitis liver cancer, so you can use them as a liver cancer model. And I thought it was so cool that we had like this naturally occurring spontaneous, model. So it's cool that they have a dog, Duchenne's with golden retrievers too. That's actually really interesting.
Yeah.
Okay. So there have been a couple of different questions that have come through. So maybe we can take a moment to do that before we move on to the next section, which I think would be, and the next section here, I think would focus a bit more on I'd love to hear a bit more about your early stage results, some of your earlier trials, and then also what it kind of looks like if there's a rare disease family or some sort of group that's interested in working with you guys, kind of getting an idea of what you guys are looking for. So before we get to that, just a question here from Julie. Thank you for your patience, Julie. Does Genethon have access to its own GMP manufacturing facility, or is GMP manufacturing outsourced? Great question, Julie.
So we have we do our process development in house, and then we have our there's a kind of an ecosystem that was created also thanks to the IFM Telethon, where we are located in the south of Paris, about 25 kilometers south of Paris, and where there is also a CDMO for gene therapy. It was linked to us, to Genethon. Genethon had this whole GMP, spun off, it became a CDMO. And today it's very large with capacity, and so it's literally across the road, which sometimes is interesting in terms of tech transfer when you are close by, and so that's our GMP facility.
Gotcha. Excellent. Okay, and then there was one other question just coming in. Thank you for everyone who has put questions so far. If you have some more questions, feel free to put them in the chat. We've had people from all across the United States, from Great Britain, India, Brazil coming. Thank you for coming. Here's another question coming in. Can you elaborate a bit more on the stability of these products and how to maintain supply chain? So I guess, for when you're developing AAV gene therapy doses, I know this is a bit more of this isn't specific to Genethon, but like what does storage and stability look like for the gene therapies that you're developing?
So yeah, I mean, this is gonna kind of get a little bit into the technical side. So you have to maintain obviously the cold chain, and so, but this is we use a specific organization that can do that, and they can help us to deliver the product in the clinical sites. They can be, one of which is in University of Florida, and the other one is scattered around Europe. So we have there are organizations that specialize on that, and we use them. They're CRM that specialize on that.
Very good. Okay, so moving on just a little bit to, obviously really exciting to hear the results you guys have seen so far with LGMD and with DMD. Is there any other like earlier stage stuff, kind of like pre-IND or just about to enter the clinic, that you're able to share with us today? Some interesting stuff that we should be looking for above and beyond what we've discussed already.
Yeah, I mean, there's one maybe preclinical I can speak about. So we have another one, another program in another limb-girdle muscular dystrophy. This one is called calpainopathy. So the gene lacking is the one that allow us to have a protein called calpain. Again, this is much more frequent than the other. This is the most frequent of the LGMDs. And for that, we actually, so this is a bit of a second gen. So it was developed a little bit later than the other. So it does have a novel vector. It's not the classic serotype. It's a vector that has a much stronger, it did target significantly the liver. And also he has interest in, he has the transgene, of course, and it's a feature in the transgene that allow the better allow the functionality of the virus of the product. And so we have done all the clinical of the preclinical package. We did the talks. We have a very nice safety margin, and so we are ready to start all the preparation for clinical trial. So there's another interesting project we work on that maybe we can mention, and this is it's interesting also because it's an interesting collaboration that we are having. It's a partnership that we're having. So one of the issues with AAV is the fact that you have a part of the population, about a third, that have antibodies, so you cannot treat them with the AAV. This is, you can imagine, something that we've been trying to solve, because a third is a large population. And so we have a team dedicated to immunology in-house that is looking at different strategy to try to overcome the issue. And we have worked on a specific protein. It's called IDES and is of bacterial origin. And it's like a Pac-Man. It's like a Pac-Man. When you inject it for a very short period of time, it eats all your antibodies. And so you deplete your antibody, and then it doesn't last very long. And so once the effect is over, then your antibody goes up, because you do need antibodies to protect yourself from infection. And so we were the first to actually test whether it's used in organ transplantation. We were the first to see whether it would work in gene therapy. And it does. We had some interesting preclinical data. And so then we signed a collaboration with the Swedish company called ANSA that is commercializing the product for organ transplantation, liver transplantation. And so we had, so you have to be able to, we wanted to test whether it works. You could take a patient that is not eligible for gene therapy, that has antibodies, treat with IDES, and then see what happens. And for that, we had the perfect disease model. It's a liver disease called Krieger-Najjar. It's very, very rare, less than 1 in a million. It's a little bit like the genetic jaundice. So you have sometimes baby become yellow after they're born, and then everything goes fine. Here you're yellow for the rest of your life, basically. You don't have the enzyme that can destroy bilirubin. And when bilirubin is high, it's toxic. And the only treatment you have is phototherapy. And you have to spend like half of your life under a lamp when you have this disease. 10 to 12 hours a day. And we have a gene therapy that has been shown to work against this disease. So we took a patient with antibodies. We treated with this little Pac-Man for like 2 or 3 days before treating with gene therapy. So the antibodies were clearly decreased dramatically. We injected our gene therapy. It happened a year ago, and this patient is still not needed to use for therapy. So it worked, and it's safe.
So just to understand, just to clarify, so you are developing an AAV therapy that gives you the genetic instruction for the Pac-Man, or the Pac-Man is a separate therapy? That's
it's a separate. That's why we had the collaboration with a company that is commercializing the product for transplantation. So you have to pre-treat with the Pac-Man. The Pac-Man kills your antibody for 2 or 3 days. When you monitor, when you are low enough, you inject. And we were the first to publish by pre-treating. We published the ESGCT last year. When you treat a patient, they shouldn't have been eligible to gene therapy because the antibodies were too high. With this little Pac-Man, then you treat it, and the patient is doing very well.
Wow.
I'm shocked that doesn't already exist. Like anti-drug antibodies have been a problem for a long time, for way more than AAV. So that's actually like, there's no like quenching technologies that have been developed that are used. I'm not really in that space.
So many attempts, and probably the first problem is that. The second problem is that once you receive gene therapy, you're vaccinated. You cannot be treated anymore. That's why your durability question, Jeff, was very good, because you cannot re-inject today. And so the second now challenge will be, can we find solution to, because that the antibodies level is much higher, so the challenge is much higher, and the potential safety issue are much higher if you are not careful. So that's something that we are actually expanding our immunology team, as this is really an important today challenge that we have for gene therapy.
Okay.
Now, was that a project that you guys developed internally? The Pac-Man.
The Pac-Man existed already in transplantation, in organ transplantation, and we were the first to test it in gene therapy.
Oh, okay.
Gotcha. And so is this Pac-Man like specific to like neutralizing antibodies for AAV?
Absolutely. Neutralizing. No, it's antibody. That's why you can use it in transplantation. It's an enzyme.
For any antibody.
It's any antibody. Exactly. Okay. Sorry, and it's an enzyme that is a bacterial origin, and it cleave basically your antibody. So you need time until the antibody are generated.
Gotcha. Okay, so
you don't want to permanently depleting antibody, not at all. You really want that window. That window to get the vector in, do what it has to do, deliver the gene, and then the gene will, do what it has to do.
Okay, so it's a very wide acting. So you have to be like you can't go out living your normal life. You're getting monitored, and you're kind of like in a hospital environment. Your immune system is very deliberately kind of being like turned down.
Exactly. Shut down, and it's only 2 or 3 days. The window is very short.
Gotcha. So it's kind of like the gene therapy version of lymphodepletion for cell therapy. Only this is like antibody depletion.
Exactly.
Cool.
And so as I said, we had the presentation at the European Cell and Gene in October, when this data were presented first time. So very exciting.
Fantastic. Okay, we are approaching just the final little section here. Angela, this has been an amazing conversation so far. I guess the question would be, right, if I am a patient advocacy group or I'm some sort of foundation, we don't know where in the world to start. We have an idea for a gene therapy. We would like to work with you guys. What are kind of the standard criteria that you guys would be looking for in order to work with an organization?
Yeah, that is an excellent question. So the world of rare disease is extremely wide. There are hundreds of different rare diseases, right?
Yeah.
And so the first point is, and we have a certain type of expertise in house, but obviously not all type of diseases. So the first thing is to understand whether we can bring any added value to that development of that product, whether we have the right expertise, the right model, the right knowledge, right? But we do have a very strong. So the IFM Teleton does many beautiful things, including in having his own call for project and has invested a huge amount of funds into many projects in rare disease, gene therapy, small molecule, all around the world. So there is a very large network of expertise, and sometimes we can direct the family through the right PI, through the right hospital, through the right organization, where that particular disease, where there's expertise in the particular disease. The expertise on the disease is key.
Gotcha.
Yeah.
Okay, so yeah, you're really just looking at your internal capabilities to be like, have we done this before?
Or do we know, or is it part of what our expertise is like today? And the expertise is evolved. As I said, we started lentivirus, immunodeficiency, and blood diseases, and then we move as the AAV became more and more. They kind of became more standard of care. Then we moved to AAV, and that's where we started to focus on the muscle and the liver.
Gotcha. Super cool. I guess the other thing here with regards to where the future of gene therapies are going, and again, obviously you guys have loads of background and have been very fundamental to the success of gene therapy so far. I'd be curious to get your thoughts as to how you guys are viewing the industry evolving into the future. Do you see AAV continuing to be dominant? Are you guys looking more into LNPs now? I'd be curious to get your thoughts on where you see that shaking out.
Yeah. We are looking into LNPs. We look at the gene editing as well. We have a team dedicated to gene editing. LNP, for what I'm told, will be difficult to substitute viral vector when you're talking about delivering DNA. Mm-hmm. So but we are very interested in building our presence in LNPs. We think that gene therapy, there is probably a need to maybe work on a more targeted approach, so reduce the dose, increase the safety, and open up to more frequent disease. And that would be an interesting way of getting the gene therapy where, do you remember when the monoclonal, maybe not because you're quite young, I remember when the monoclonal antibodies started to be developed, and then, were niche, and then, as you start to decrease dose, decrease cost, decrease everything, they became very common. So gene therapy probably has to go to the same way. And in that sense, so becoming more targeted, safer, lower dose, and reducing the manufacturing cost to make it more affordable, to make it more available to broader type of diseases. I think these are the two probably interesting direction. And find alternative way of delivery. They are safer, of course, and as efficient as, because as I said, the virus was designed by nature to deliver DNA, so it's difficult to deliver genetic materials. It's difficult to find something better. But LNPs is certainly an interesting promise.
Mm-hmm. Makes sense to me. Yeah, I somewhat agree. Right, the challenge of LNP so far has just been the delivery mechanism that AAV has is very unique and somewhat difficult to replicate, and the ability for it to exist as an episome, it's been difficult to make that work inside of the LNP space. So yeah, I don't think AAV is good.
There's a lot going on LNPs. I was surprised that last year in New Orleans, American Cell and Gene Therapy Society conference, there was so much going on LNPs. So it's a very hot field at the moment, very promising.
It sounds like you're saying that ASGCT is a real happening conference, Angela, and that anyone who's worth their salt inside of this industry should be at it.
I prefer pepper, so.
I feel like you're having a conversation again, right?
Excellent. Well, thank you so much for coming on, Angela. We appreciate you spending a little bit of your evening with us. It's just around 1 o'clock our time, but it's right at supper for you. Thank you so much for coming on. We'll probably thank you everyone else who is tuned in and for coming today. Thank you for your questions, and we look forward to seeing you next time. We'll get you to stay on for just a second, Angela. Don't go anywhere. Yeah. We'll end the stream, and then we'll debrief after. Thanks for coming, everyone. Cheers.
Thank you.